trend doesn't always hold up. So speaking of that, there's a very common regenerator like we heard in our last presentation called the axolotl that can regenerate its limbs and many other things. But it has an interesting globin environment within its genome in that it has 20 hemoglobin genes and no myoglobin genes whatsoever. So very closely related to the axolotl is the lungfish and the Polypterus, both of them having also very interesting globin environments and not the same among them. So I'm really wondering specifically why are three closely related species having very different oxygen management strategies, and then looking to more distantly related but still a regenerator, the zebrafish. It only has one myoglobin gene, but luckily zebrafish have been awarded with many CRISPR and genetic editing capabilities, so that's why we're including it in this study. But yeah, largely very different oxygen management strategies. So we wanted to focus specifically on Polypterus and lungfish within this study because they're doing some interesting things with their myoglobin genes that I'll talk about a little bit later. But largely, it doesn't matter as much which globins they have, but which are expressed, and which are expressed differentially between homeostasis and regeneration. So there's a couple of ways we analyze gene expression. Like Isis mentioned in her presentation, we can use spatial transcriptomics, which all you really need to know about this technique is it's going to give you a sense of the spatial expression along the physical fin. And each dot is representative of a collection of cells. So this is not just one cell. And the expression is obviously based on the darkness of the color. So darker colors indicate higher expression. But it really lacks that single cell resolution that we're looking for. So it will give you some information on where within the limb expression is happening, but not really to the single cell. So we have another, one of the presentations doing that, but we have another method, single nuclease RNA-seq, which Isis talked about a little bit, which will give you a sense of the nucleic RNA expression quantitatively and clustered by cell type. So it will tell you which cells these genes are being expressed in and how much expression is occurring. And then we also have bulk RNA-seq, which can give you just a normal number of the cytoplasmic RNA expression over 2 regeneration time points, so either homeostasis or generation. And ultimately, we wanted to combine all of these methods to get a much more informative sense of what's going on with these myoglobin genes. So we're utilizing HCR fish to do that, to really get a more specific technique to really understand that expression fully. And I'll just explain a little bit about what HCR is. So in simple terms, within the physical tissue, you're able to mark expression of genes with fluorescent tags. So when I show you my HCR, you're going to be seeing lots of pretty lights flashing up on the tissue, and that is where genes are being expressed. So there is a method where we have a probe set that goes into the tissue and binds to an RNA target of interest. In this case, it would be myoglobin. And then there's a thing called hairpins, which have fluorescent markers that are notated as stars here. And there is an amplification step. So the hairpins go into the tissue, bind to the probe set, which is then bound to the RNA target. And then there's an amplification where hairpins are binding to other hairpins. So the fluorescent signal is going to be amplified to the point where that chain reaction is occurring. So this is not yet HCR fish, but I wanted to give you a little bit of sense of where we are orienting you when we show you the HCR. So this is 2 Polypterus fins, one at 0 days post-amputation, one at 3 days post-amputation. And what you're looking at right now is H&E staining, actually. So anything in purple is going to be something acidic, so cell nuclei, or you can tag cartilage and bone, so that purple line running through the middle is bone. And anything in pink is going to be like cytoplasm, muscle, collagen, those types of things. And those that black box, like in the middle of the fin, is just going to give you an orientation whenever I show you the next slide. So this next picture is going to be within those two boxes. All right, so here's what we're looking at. So once again, 0 days post-amputation, 3 days post-amputation, and we have 2 myoglobin gene duplicates that we've marked with fluorescence here. And it looks to me like MB1, which is just a notation that we gave it, 1 myoglobin gene duplicate is expressed largely in the muscle cells, specifically on the outskirts of each muscle cell. And then there's MB2, which is expressed, we believe, in endothelial cells as their blood vessels are running through the muscle. That myoglobin is expressed in those cells in particular. And DAPI is going to be the blue dots that you're seeing. Those are just staining cell nuclei. So it's a lot easier for us to zoom in even further than this picture to be able to see physically within the cell where is the fluorescence popping up, which is something we can't do with all the other methods. And so we can look at everything at the same time, and we can also multiplex 2 genes onto each other and view them at the same time, which is really awesome. So here, myoglobin is behaving mostly what we would have expected. It's in the muscle, not really a surprise there, but we do have some instances of it behaving non-traditionally, and that's what I'm going to get into now. This is going to be 3 days post-amputation polypterous fin, specifically within the regenerating skin. That's what you're about to see. Very non-traditional. So MB1 once again is in green. Keratin 17 is just the basal epidermis marker, so the innermost layer of the skin. And we do see some green popping up in almost every skin layer with the exception of the basal epidermis. So we found that to be very interesting. Myoglobin is behaving very non-traditionally here, especially during regeneration, which is even more interesting. So I'm wondering why these trends are happening, and what do these trends say about the overall oxygen management strategies within these animals, and are they utilizing myoglobin for some purpose to deliver oxygen to the regenerating skin in some way? Those are the big main point questions that we're really asking when we see these. Okay, so now I'm going to show you a lungfish fin, and this is specifically going to be the skin and muscle in a non-regenerating area for both 0 and 3 DPA. So right here, these are 2 completely separate myoglobin genes, so MB3 and MB4 within the lungfish fin. And you can see at 0 DPA, they're both expressed in the muscle. The MB4 is expressed in the skin as well, but they're each expressed in areas of the muscle that the other is not, which I found interesting that there's some sort of specialization going on here that is different from the last HCR I showed you, where it's kind of a different kind of specialization. But then during regeneration, it seems that MB4 kind of consolidates itself to the skin instead, and MB3 remains in the muscle. So that's just a really interesting, non-traditional behavior that we're, seeing with these myoglobins. They're not. It's kind of like a breaking expectation of what we thought myoglobin would be doing. Now I'm going to show you a regenerating zebrafish fin. Specifically, can't really see here. This quality is bad, but there is some expression that we're seeing in the zebrafish fin. Although more work is ongoing for this to really fully be able to interpret this. I have an experiment lined up to do some whole mount HCR, so doing HCR of the entire zebrafish animal to really fully consolidate this information. But I wanted to say that the zebrafish fin is mostly made of skin and bone, no muscle at all. So it's very interesting to see any myoglobin expression whatsoever. And then, in addition, we got hold of a transgenic line that has no myoglobin gene in their genome whatsoever, and we want to do a regeneration assay to see if they can still regenerate without having myoglobin at all. So overall, investigating these non-traditional globin gene expression patterns is going to help us categorize the oxygen environment within the fins that are regenerating, and then also better inform the functional requirements that are needed for regeneration to occur, specifically considering oxygen in that case. And that's just one piece of the puzzle to unlocking the overall mechanism of regeneration that can be applied to human regenerative medicine in the future. So I'd like to acknowledge my beautiful lab members and my mentors in the Lanyap program as well. If you'd like any really cool fish photography, like I utilized in this presentation, I'm gonna shout out my co-mentor. And I'm open for questions.
What do you think's going on with the zebrafish fin? Why is myoglobin popping up?
Yeah, that's a good question. So like I said, the zebrafish fin is made of mostly bony rays and skin. That expression that we saw could be indicative of blood vessels running like proximally to distally. Like we saw in the Polypterus, there was a myoglobin expression within endothelial cells, so with blood vessels. I think that could be the case, but like I said, there's more work to be done to really like emphasize what's going on here truly, the biological reality of it. Yeah.
Have you given the sequences of those proteins out?
Yeah, so actually, this past week, I was generating some protein sequences for those genes, and I'm going to do an analysis of what is the difference between those two protein structures to really see if there's like any difference